Hello, everyone. Thanks again for joining us at the Competitive Advantage Talks presented by Kager, also known as Craft Analytics Group. My name is Patrick Sherry, and I'm an MBA student here at MIT Sloan. It is my pleasure to introduce this fantastic presentation of mice and hockey players, cross-disciplinary lessons about tracking data. Please join me in welcoming our speaker, Margaret Cuniff, data scientist at the Carolina Hurricanes, to the stage.
Thanks so much. Hi, everyone. Thanks so much for coming, and thanks so much to the organizers for inviting me and for all the work that you did organizing the conference. It's great to be here. So a little bit about my background, where I came from, and where I am now. So two of my great passions since high school have been hockey and the brain. I did my neuroscience, I did my undergrad in neuroscience here-ish at MIT, and was also thrilled to live on a dorm floor that had a very low skill, high enthusiasm intramural hockey team. So my major was neuro, course 9, but I also learned how to skate and learned how to play hockey here. I went on to do my my PhD at UCSF in neuroscience. Played less hockey there, but still followed it closely. As you can see from this picture of my recording rig, where I'm actively, there's a little brain slice somewhere in there, and I'm recording a neuron, and also have the Stanley Cup playoffs off to the side. And so, it was an ongoing part of my life, something that I've been interested in a long time, but never had really thought at all about doing sports professionally. Mostly because it never crossed my mind as an option for someone with sort of my skill set and my niche and the kind of things I was interested in working on. And so it was less that I'm not interested in it than it didn't compute as something that I could or would do. But that changed with the introduction of player and puck tracking to the NHL. And so after finishing up at UCSF, I am now a data scientist with the Hurricanes. So why did the player and puck tracking change things? So I was doing experiments in grad school where I was recording from multiple parts of a mouse's brain as they ran around an environment. So it's this situation where we have multiple channels of complex data that are both meaningful on their own, but also in combination. And the position of the mouse in space and time matters a lot for the interpretation of the data. And so when they announced player and puck tracking, I was thinking about the stuff I was currently working on and was really excited about all the opportunities that this would open for hockey. And it's that's kind of what I'm doing right now. If you think about multiple areas of a mouse's brain versus multiple hockey players and different areas and mouse mazes versus the rink, they're basically exactly the same thing. And so I for the first time was hey, this seems like something I could maybe do. And I was lucky enough to get an interview, and my boss, the now GM of the Hurricanes, Eric Tulsky, was the kind of person who also was at least, hey, that sounds like something familiar. And those things actually relate, which I don't think there are many people in the NHL who were necessarily on the same page as us, but we were on the same page. And as much as I say that there are similar things, my pitch in the hiring process was less oh, I have these algorithms that are going to be ready to go on day one because this is exactly the same problem, and so we've solved it because they are very different. It's not exactly the same. But a lot of grad school is basically having this chunk of time to try and figure out a novel problem. And it curates a type of approaching pathological stubbornness where you're trying to figure out how to do something someone hasn't done before, and essentially going towards questions and saying, okay, how do I answer these things? And often with tools that you maybe don't have yet. And so, as my project evolved through grad school and, the different types of experiments I did changed, there were things where it's okay, here's the next question I'm interested in academically. How would I answer that? It's well, I guess I have to learn this new technique, or I guess I have to find this other thing. Like, I don't really know how we would do this, but that's the direction I want to go, and I'm going to figure out how to get there. So, when I was first talking to Eric, I was basically saying, like, okay, I've worked with similar data. I know what the landscape is like. We're getting this brand new data in the NHL. It's wide open green space, and we're going to have to figure out like what to do with it. And I know that you can probably hire someone else on day one who has better like data scientist skills. But I know my training is in looking at problems and saying, hey, I want to solve this. I don't know how today, but I'm gonna figure it out. And so I think that's gonna be something that could work in hockey as well. And again, luckily that worked, and here I am. So I'm going to talk a little bit about a couple sort of recurring themes and why these things are similar, and the type of things that I think about that I sort of learned to think about in grad school and now still regularly think about when dealing with hockey data. And so one of the first things that many people have brought up in many talks in this conference is just like, how do you get measurements out of these complicated data sets? Because at the end of the day, we want to have insights and we want to report things, and you just have this like pile of data, and there's something interesting in there, but getting it out is non-trivial. So we're briefly going to talk a little bit about mice. I promise I'm not just going to like pivot to my thesis talk, but for any of my examples to make sense. I studied anxiety, and mice, as you know, are very timid. They're prey animals. They're very cautious little guys. But they also they forage for food, and they have to go out and exploring. So there's this like real competing drive in them between novelty and exploration and safety, because they don't want to get caught by a hawk, but they also can't just sit like in a hole all day and hope that something appears. And so one of the ways that we can study anxiety is to sort of take advantage of this natural drive that they have and put them in situations that are sort of more or less anxiety-inducing. So this is something called an elevated plus maze. It's a plus that's off the ground by a couple of feet. Very straightforward naming here. And for a mouse, that's like really high in the air. And you have two of these arms that are closed, and so they're nice, they're shaded, they're safe, the mice like them, and two that are wide open, and we do them under bright lights, much like I am now. And the mice are not into that. And so normally what you see is here the horizontal is the closed arms, and the vertical is the open. And so most mice will kind of like stay most of their time in the safe areas, and like they'll go out a little bit and sort of explore again. They want to know, like, is there something out here? And once they realize, like, no, there's no reason for me to go out there, like, they're back. And so, in sports terms, think of this somewhat, like a goalie. They're like pretty well tethered to the goal. Like they'll, go out on occasion, sometimes more aggressive, but it's a relatively conservative position often. But then I worked with these mice that just were fearless, and they went out and freely explored and didn't seem to care, and would go and peer over the edge, and were not at all daunted by this. You do also get this with goalies sometimes. So this is the Hurricanes' other goaltender, Kochekov, who is a great goalie and has very little fear in a way that sometimes causes fear in the press box for me watching. But always exciting. And so, one of the things when working with, behavioral data in science or, observational data in sports is you only have so much control. And, this is from a hockey standpoint, very jealous of baseball. But, you can't run more trials or force situations to come up more often. So, I said we take advantage of the fact that these, mice like to explore, and so like, oh, we can look at them in safe and unsafe situations. Sometimes you'll have mice that just want to like sit in the corner, and okay, that like kind of really limits what we can do with that data, but also we can make them go out. And I can't ask the coach like, hey, can you shuffle the lines so I have like a better control set, or like this guy doesn't really have enough ice time, so can you deploy him because my sample size isn't big enough? He would just not respond. I think it wouldn't even, it would just leave. And there's also just a ton of confounds that you can't eliminate, when you have all the different players on the team, you have the other team, maybe you're interested in looking at how a player performs on a breakaway, and then they go up against a goalie who's going to come out 20 feet from their net and challenge them, and that's not a usual situation like the rest of your data set. And it's somewhat glib, but also true that like the first stage for a lot of these things is just, all right, this is what I have, and this is what I have to deal with. And it's very easy to sort of waste time and energy on, it would be great if this were true, if this data looked differently, or if I could do this control or whatever. And it's fun to think about like blue sky things, so not like, oh, what the best case scenario is, but it's not helpful to sort of dwell on like, well, if only X, Y, Z. And so instead thinking about like, all right, given these bounds, given the things that I have to deal with, what can I do with this, and what do I have to do to make this meaningful? One other thing is hard is figuring out how to find, again, like features in this messy data set. So this is a recording of a single neuron firing 5 action potentials. This is what was happening in that picture where I was watching the Stanley Cup on the side. This is what I was actually supposed to be doing. And most people in this room probably don't know like what the names of things are, but you could pick out features in this, right? Like if I asked you like, hey, can you like, put numbers to this? It's like, okay, there's like 5 lines, they're this tall, they're this far apart. Like, there's an intuition for like what it could look like to go from this raw data to numbers. And we can think of this as something like shot locations, where again, like there's an intuitive meaning, we can have a sort of handle on it, but we also don't have the whole story in either case. And so, the recordings that I started doing with these like multi-site examples were, they're minutes long, these are happening while the mice are running around. We're interested in both these 2 recordings on their own and also how they relate to each other. There's lots and lots of lines instead of just a couple lines. And so it's harder to know like, all right, what is in this? And that's similar with the tracking data where it's like, all right, we know there's a ton there, we're interested in it. You have a player skating around for a game, like what does that mean? And it's hard because like when you have these huge piles of data, it's really easy to go on some fishing expedition and find something that's correlated but like doesn't necessarily mean anything. And one of the things that again comes up in research a lot is that breaking new ground and doing new research doesn't mean that we have to start from scratch and start from zero. And so one thing as far as breaking down my data, as you probably heard about brainwaves in the context of like sleep, like slow wave, REM, et cetera. So we know that there are different frequencies of brainwaves that are important for different situations. And so, we know that theta frequency matters for this region I was studying. So instead of looking at like the whole mess of the data, we can just take out this one frequency, and now we have this like much cleaner set. And using tools, that signal extraction is coming from, stuff that neuroscience has stolen from electrical engineering and other signal processing fields, not reinventing that wheel. And, we can think about domain expertise to meaningfully break down the data. So, again, in the context of anxiety, I have 10 minutes of mouse doing whatever it wants in the maze, but like, we're really interested in why this one set of mice chooses to go and spend all this time in the scary areas, and these other mice are much more conservative. So we can look just at the point where they are in the safe space and they're approaching the open arm and making a decision, because that's when they're thinking about sort of the choices that that are relevant to us. And so this could be something like the not starting from scratch would be, in hockey we can look to soccer, we can look to basketball. There are advantages of being a little bit behind the curve. And meaningful situations, we don't look at a whole game. We can look at like, the seconds leading up to a shot. We can look at time in the defensive zone, sort of taking chunks out of this. And by sort of constraining your data in these ways, you can get to much more interpretable results, than just if you go on these like fishing expeditions. So in this case, basically, we saw that with the bold mice who are not afraid, that there was a very different, there was a big difference in how their brain behaved at this frequency that we knew was important in this brain region at the point that they were making the decision of, am I gonna go somewhere safe or scary? So we've both broken the problem down to something simpler, and in doing that, we have gotten to something where we can have a much better handle on like what this could mean. And part of that is even when you've broken it down and you're like, okay, we think we know what this means, like how do you have confidence in your results, and how do you think about these things critically and be able to sort of stand behind your work, and how do you identify things that maybe aren't quite what you think? And one of the big things is that you can't eliminate all uncertainty. Like even if you've done this thing where you can like, oh, we've really constrained this data. We're no longer looking at a 60-minute game. We're looking at a very tiny chunk. The other team is still there. Or if you're looking at like defensive pairs, you can do like with and without you stuff and try and separate like, all right, if we look at all these combination of pairings, like what is one guy's contribution versus the other, but you're still always looking at pairs of players. You might just be looking at different pairs, and there's the quality of competition. Like there's, there are things that you can and should do to try and again, like constrain your, the bounds of what you're looking at, but like you're not gonna end up going from this like wild, unconstrained, messy data to something like perfectly clean. And the point isn't like, okay, so you shouldn't do anything, because that's a very useless talk and also very boring. But, it's this balance of you don't want the perfect to be the enemy of the good. Like 100%, if, looking at something and say like, well, we can't account for this shouldn't be the end of the story. But it also means you need to be clear-eyed about sort of what the limitations are and what trade-offs you're making and sort of what choices were on the table. And so, a lot of the way that I think you can prepare for that is by having like a really strong critical practice. And I think this is something that academia does well in a lot of ways, where just very regularly you are critically examining your work, others' work, and learning how to question things. A big thing in grad school is we would have journal clubs where you'll read papers that another lab published and do sort of like a very close read with the lab. And part of it is you want to see what other people, like what their findings were, like keep up with the research in the field. But when I started as a young grad student, it was like, all right, so that's what they said they did, and they did that, and it's great. And the sort of skill learned through grad school that my advisor was a great model for was really looking at something closely and saying like, all right, but like, could you interpret these data a different way? Like, this is what the story that they're telling, and we've heard a lot about data storytelling this weekend, and that's certainly something in academic papers as well. And so, this is what they're saying, but like, does it have to mean that? Like, do their controls actually eliminate that problem? Is there a different experiment maybe we would like to see from them? And really digging into like, all right, what exactly is happening here? And not in a way where it's like, oh, we're trying to like tear apart this work and say it's not good, but like really sort of nuts and bolts, like what's the storytelling versus what's the reality, and how might we do that differently? And regularly presenting to different audiences is a big part of, again, academic life. You're giving lab meeting to the people who are working on like basically the same thing you are. You're giving like presentations to your department to people who are similar but not quite identical. Going out to conferences and stuff, and sort of really cultivating this practice of hearing from people with different feedback and different expertise. And certainly this is huge in sports with getting feedback from different people in different parts of the organization. And like even in early stages, we can talk to coaches and other people who are might not have thoughts or opinions or want to comment on like the statistical methods, but they have good intuition for like, is this an interesting question? Do we care about this? Like, hey, you're not accounting for the situation. And giving and receiving critical feedback is a skill. It's hard, both in terms of like how do you give stuff that's interesting and actionable, how do you like not take things personally. There's a reason grad students all talk about hating committee meetings, because like it's not fun to stand up and have someone like really like tear your stuff apart, but that makes stronger work in the end. And I think that's really critical in this field where, again, a theme that comes up in many talks and panels this weekend is that good analysis is nothing without good communication. We're trying to do things that empower the team. We're not trying to just like publish a paper on hockey and say like, hey, we found this cool thing. We want to do things that people actually use. And having this practice where there's this delicate balance between we deal with uncertainties and you want to communicate your findings, and you want people to use their work, and you also don't want to oversell it, and you don't want to lose faith. And having a really deep understanding of what you actually have and what the limitations are really shapes your ability to do that and to be able to know how to communicate and know what's important and not. And also, it's a way to build trust and communication for, trust and foundation for effective communication. Like, if you go up to coaches and show them something and they say like, hey, but what about like, did you account for this situation? And even if the answer is no, if you can say like, yeah, that's like, good point, that's like a problem that's here, like, here's how we've thought about it, here's how like we are trying to do it, we haven't solved that, but like, showing that, we know we don't have all the answers, and we're not trying to say we have all the answers, and, being realistic about, having people with domain expertise buy in and provide feedback. It's important to have our strong basis, which we can get by like really being in the weeds with our own work. And so unfortunately, like there's no one weird trick for figuring out how to work with any of this data. Sorry if you hope there was. You should know by the sports that are like way more established than hockey that haven't figured it out, that certainly we haven't either. But with that, would like to thank everyone I work with at the Hurricanes, and also, again, my graduate work, and particularly my advisor, Picasso Hall, who is a big basketball analytics guy and was very suspicious when I started talking about leaving academia until I said I wanted to go into sports analytics, and suddenly he was like totally on board. So thanks for your time.
We have time for a couple questions.
Hi, Margaret. Thank you for your time. My name's Josh. I was curious on your opinion. Like, are there any specific areas in hockey or in the NHL where you think analytics still has like a lot of room to grow? One in particular?
Yeah, I mean, pretty much everywhere, which is a little bit of cop-out, but as I said a couple times, like NHL is relatively far behind other teams or other sports, so we're still like in our infancy of data. The thing that comes up with any sport that's getting tracking data, but it's certainly true in the NHL, is defensive play and off-puck play. We have great or increasingly good rich event data. So being able to do I showed a shot map, and being able to go from like shots to like all puck touches was like a huge advantage, but that still leaves everything that happens off puck. And so I think that's really sort of one of the main spaces that really is just fully not possible pre-tracking data.
Time for one more question.
Hey, how much do you reference the neuroscience background and some, I guess, when you're making these analogies versus just talking about the conclusions in terms of the actual hockey analysis? Do you try to give the full analogy and where you've come from, or do you just try to focus on the actual conclusion that you've come to?
I mean, by the time I'm talking to like downstream stakeholders, just the conclusion. They don't care or aren't interested. Certainly earlier in the process, it'll come up with like, there are things where I've, if I'm talking to like Eric about like, hey, I was thinking about this like, type of recording we would do or type of measurement that we would do. And like, I think, there have been times that I've had like slides where it's like, all right, I haven't mocked this up with hockey data yet, so here is a neuro paper because, I know that he understands what I'm saying as far as that being a transition, but that is a very particular audience, and certainly by the time we get downstream, the main takeaways are often divorced even from the details of the stats, much less the sort of origin of my thought process. So I don't actually compare the hockey players to mice in front of them, but cool. All right, thanks so much, guys.